Armstrong, and we're going to be doing another in the series, Science for Today. Now, this is a public lecture series which is initiated by the Faculty of Science and Technology at the University of the West Indies, Mona campus. Today, we're going to be talking about an animal which weighs only 2.5 milligrams. I bet you don't know what I'm talking about, right? Because you did not read the flyer. Obviously, I'm speaking about the mosquito. Now, this animal is considered the deadliest animal in the world. For some people, when you're asked, which animal do you think is the most deadly animal in the world? You may get responses like the black widow spider, or a lion, or the black mamba snake. But in fact, the mosquito is the deadliest animal in the world. And it is so because the mosquito is responsible for over 1 million deaths worldwide. Now, right now in Jamaica, we are experiencing a dengue outbreak. So our presentation today is actually quite timely because we need to know more about the overview, which will be provided by Dr. Karen Webster-Carr of the Ministry of Health. We'll also talk about some clinical evidence for some new ways of treating dengue using papaya leaves, and that will be done by Dr. David Pickey. Dr. Sheena Francis will tell us about insecticide resistance, and you and I will know that when we try to spray these mosquitoes, they're still flying around and biting us as if there's nothing. They've had nothing, it has no effect. So Sheena is going to talk to us a little bit about that. And then we will close off with Dr. Johan Anton. And Dr. Anton has been so kind. He quickly sat in for Professor Charles Grant, who had an emergency and had to address that emergency. So he's not able to be with us. But we thank Dr. Anton, who we think is an excellent replacement for Professor Charles. So without further ado, I'm going to introduce Dr. Karen Webster-Carr, and she's going to present for us the overview. Now, I should have started by saying to you, we have a few house rules, and one house rule is that if you have a question, please, as you remember it, just put it in the question and answer section so that we can address it at the end. So how we're going to do it today is that we're going to have each person present, and then we'll open up for discussion. So I'm going to start now with Dr. Webster-Carr. So Dr. Karen Webster-Carr is the principal medical officer and the national epidemiologist of the Ministry of Health and Wellness here in Jamaica. She has been in public health practice for over 20 years and spent most of her time at the local health department in Kingston and St. Andrew. While at Kingston and St. Andrew Health Department, she led the response to the large urban outbreak of malaria. Now, ladies and gentlemen, at the time, Jamaica had not seen a malaria case for over 45 years. And I think you might agree with me that this introduction is timely because we're talking about mosquito-borne diseases, of which malaria is one. So successful was Dr. Webster-Carr's strategies that we were able to re-eliminate malaria from Jamaica. She leads on national health programs and policies, especially as it relates to epidemiology and research. Dr. Webster-Carr is the chair of the Health Thematic Working Group for Jamaica's National Development Plan. And our development plan, if you don't know, is called Vision 2030. She is principal investigator for the Centers for Disease Control and Prevention grant. And the grant looks at surveillance and response to avian and pandemic influenza in Jamaica. And she's a co-principal investigator for the Jamaica Health and Lifestyle Survey of 2016-2017. Dr. Webster-Carr was critically involved in Jamaica's response to the COVID-19 pandemic. Many times we'd see her on TV giving us updates and telling us what was going on. And her role really was to provide information for action. And I think her unit did just that. Dr. Webster-Carr chairs the Essential National Health Research Committee, And this committee focuses on research for development, and right now they're working on a program to advance the national COVID-19 research agenda. Ladies and gentlemen, please join me in welcoming Dr. Webster.
Thank you, Dr. Gordon-Strong. The presentation might be shorter than the introduction. Thanks. So what I'm going to do is just go through an overview of dengue. And dengue is not new to Jamaica. Dengue was first described as a distinct disease in 1943. And we had about 1,546 cases described. All 4 serotypes have been identified and have caused outbreaks in Jamaica. The largest reported outbreak to date being in 1977, when type 1 was first identified. And just looking on the chart on the right, dengue outbreaks have become more frequent. The number of cases in between outbreaks, some of these in between outbreaks look like the outbreaks of the 1990s. So inter-epidemic periods, the number of cases have increased, and the dengue-related deaths have also increased. And these are features of hyperendemicity. So Jamaica is hyperendemic for dengue. The yearly pattern of dengue in Jamaica shows a distinct seasonality, with the highest incidence being seen in the latter part of the year, peaking around October to November. And as this group will know, the factors that influence dengue fever and dengue fever outbreaks are numerous, and social, biological, and the introduction of the different serotypes. Analysis that have been done, there is a strong association between outbreaks and temperature, as well as rainfall or lack of rainfall, with the association with temperature being greatest. So I'm just moving straight into what we are seeing currently. And as of the 24th of October, we had recorded 2,470 cases, of which 577 we have confirmed on PCR. The serotype 2 predominates. There have been other cases, other serotypes identified. Serotype 3, including one that is a co-infection with 2, and 2 serotype 4. There have been 9 dengue-related deaths classified so far, 2 of which is confirmed dengue. So this is what our epi curve looks like, and the last 2 weeks will fill out, so the epi curve is still increasing. Week 40 ended on Saturday last. So, by the time we get symptoms and the persons seek care, we are lagging by 2 weeks. This is what our monthly numbers look like. We only have threshold for months. And so this is what we're looking like. At September, we went as much over 1,500. And so far in October, we have 460 cases. But this is probably less than half of what we will see in October, given where we are right now. So the distribution across the parishes, the Kingston as St. Andrew, followed by St. Catherine, has the greatest number of cases. But when we look at the cases per population, this is 100,000 population. That's the rate. That the figure is not in the title. So St. Thomas by far has the highest rate, followed by Portland and Trelawney. When we look at it in 2 weeks blocks, all the parishes increasing, and the expectation is that weeks 39 and 40 will also continue to increase. So the number of the cases, the rates per 100,000 population is increasing. St. Thomas and Portland continues to have the highest rate, and the rates in St. Thomas continue to increase. These are the epi curves by parish, and it basically follows a similar picture to the epi curves, the national epi curve. So far in this year, we have surpassed the number of cases we had in 2016. We are at about 25% of where we were for 2019. And so it seems that it will be a bigger than normal, a big outbreak. And I'll touch on that a little bit more. We have had over 100 persons hospitalized by Epi Week, and this data is what we have at the moment. We suspect that this will also increase. So the persons in the 5 to 14 age group have been the most affected, and followed by children 1 to 4, and then less than 1. So there, as I mentioned earlier in the presentation, there are a number of determinants for the vector-borne diseases, and in this case, we're talking about dengue. And part of this includes our socioeconomic. Some persons think it's a inner city disease, and you probably would be wrong if you said that, because in many of the outbreaks, the more affluent communities have had the higher rates of dengue. Some beliefs about mosquitoes, some people really don't believe mosquitoes spread certain diseases. And in addition, the little wigglers in the water indicates, as some believe, that the water is good and the water has not been poisoned. And so they are, that is part of it. Of course, rainfall, climate change, and temperature are important indicators, as well as what we do in the health sector, how we manage the density, or how we educate on these matters. And then there are lifestyle practices, or landscaping, or keeping up potted plants, or water features, etc., are probably important outcomes. So what are the drivers of this current outbreak? So the predominance of dengue virus type 2, we have not seen dengue type 2 predominated since 2010. And so the children born, so the 13 years and younger, would be susceptible. And this is what we see playing out in the most affected. We had, recall earlier in the summer, increase in temperature above normal in 2023. Those people who know better than me, it is said 2023 is said to be an El Nino year. And then we had issues with drought coming up to the summer, and in the summer up to now, so, and then we have increased rainfall, all set up for this outbreak? It appears because the outbreak started when on the upswing of normal dengue season, that it would be a large outbreak and go into 2024. So, of course, the Ministry of Health would respond in a way that would objective is to decrease or minimize the impact of the dengue outbreak. And these are organized on an effective coordination. So we activated the National Health Emergency Operating Center, and with that, the parish operating centers have been activated to coordinate the response. Surveillance is done on a daily basis, and we report to the public once a week. We have optimized the lab capabilities, not necessarily where we want to be for serology. We are up and running very well for PCR, and to ensure adequate case management. This includes not specific for dengue laboratory testing. The targeted public education and awareness, some of you may have seen those, and we have enhanced vector control measures. And the clinical management, because all of this is to prevent severe disease and death. So the indication of warning signs and adequate and treatment of severe cases. Just to let you know that we did vitals on dengue in Jamaica. Obviously, it doesn't have this outbreak period, but it has the previous outbreak and gives a little bit of the history of dengue in Jamaica. So that completes my presentation. Thank you.
Thank you, Dr. Webster-Carr. Thank you for providing this overview, which really included it took us all the way from the historical perspective to the current time. And we thank you for your presentation. As I had said before, we're going to go through the presentations and then we're going to open up for questions. So if you have questions for Dr. Webster-Carr, please just put them in the question and answer section, and we will read them and have her address them when we get to that point in the program. So our next speaker, and I just want to remind you of what we're talking about today. This is our topic, killing the buzz, the fight against dengue and other mosquito-borne diseases. So our next speaker is Dr. David Pickin, and Dr. Pickin is going to be talking to us about global clinical evidence that has been supporting the use of papaya leaves for the treatment of dengue. So I'm going to give you a little background on Dr. Pickin so that you will know that he really is quite well poised to speak to us and an expert in this field. So Dr. Pickin is a research fellow at the Natural Products Institute at the University of the West Indies. He holds a Doctor of Philosophy in Biochemistry from the University of the West Indies and BSc Honors and Graduate Diploma from the University of Westminster's School of Integrated Health, where he graduated as a medical herbalist. His research interests include pharmacognosy, ethnopharmacology, and ethnobotany. A primary aim of the research undertaken by the Natural Products Institute is to support the development of strong evidence for safe medicinal use, for the safe use of medicinal plants within the Jamaican healthcare system, and to support the development of successful Jamaican natural health products. He has published peer-reviewed articles, book chapters, and books, and he's presented at national, regional, and international conferences. He's a regular contributor to the Caribbean-wide TRAMIL network, and TRAMIL means Traditional Medicines of the Islands and Online Pharmacology. He completed and published Jamaica's first Tramil survey in 2011. Dr. Pekin, we welcome you to deliver your presentation at this time. Thank you. You're muted. You're muted.
All right. Thank you so much for the invite to participate today. And so let me get straight in because I know we're short of time. So I'm going to talk about the evidence of the use of papaya leaf in the treatment of dengue. I'm assuming everyone can see the screen. So dengue, Carica papaya, known also as pawpaw, tepaya, lecosa, papaya, and ibepi in different countries. It's a member of the family Caricaceae. It's a small succulent broadleaf evergreen tree that bears fruit throughout the year and that we all know very, very well. What we may not know, what some people may not know, is it's not native to Jamaica. Here in green, you can see the countries where it is native. So the native range is actually southern Mexico to Venezuela. But internationally, it's been widely introduced to countries primarily in the wet tropical biome. So traditionally, papaya, and I'm speaking specifically now about papaya leaf. So traditionally, it's been used internationally as medicine, for example, in Nigeria, for the management of diabetes, for malaria, and in fungal and helminthic infections. In Cameroon for malaria, in India as an abortifacient, and in Australia as an anti-cancer remedy. In terms of its composition, the leaves contain multiple chemical compounds. So those that have been identified to date include proteins and amino acids, including cysteine, glutamic acid, and a proteolytic enzyme called papain. Contains vitamins, including ascorbic acid and folic acid, and minerals including manganese, copper, iron, and zinc, fats, palmitic, myristic, and oleic acids. And some of the secondary metabolites that have been identified to date include, in the phytochemical class of alkaloids, carpane is the most notable, flavonoids, a number of flavonoids, most notably quercetin, glycosides, clitorin, nicotafluorine, and rutin, and then quite a significant number of phenols, including caffeic, chlorogenic, and coumaric acids. So why is it I'm talking about papaya leaf in the context of dengue? Well, there's a lot of reports internationally on the use of papaya, particularly papaya leaf, and reported effectiveness in preventing severe thrombocytopenia. So the diagram here shows severe thrombocytopenia on the left, and then someone who's taken papaya leaf and mild thrombocytopenia on the right. So what is the evidence? So what I want to do is walk us through three different levels of evidence that exist. So I'm going to give you some examples, and these aren't exhaustive. There's a lot of information in the public domain published on papaya leaf for dengue. So I'm going to go through the in vitro models. So these are test tube models, in vivo models, which would be animal models, and then talk you through some of the clinical evidence from clinical studies that have been published in the literature. So first one here is an extract of the leaf to produce carpane. And this was used in a model where plasma was obtained and platelets collected from both healthy and dengue-infected individuals. And the findings of the research was that the dengue-specific neutralizing effect on infected plasma that may exert a protective role for platelets. And that was published in the journal Viral Immunology. So another one was leaf aqueous extract, was tested against dengue-infected vero cells from African green monkeys. And this showed a reduction in intracellular replication before and after infection with dengue. And that was published in the Journal of Natural Medicines. Another leaf aqueous extract, dengue-infected THP1 cells. These are cells used as a monocyte macrophage study. And these showed significant reduction in red blood cell erythrocyte damage and hydrogen peroxide induced lipid peroxidization. And that was published in the Archives of Virology. So then jumping to research in animals. So again, with carpane, this was tested against thrombocytopenic-induced Wistar rats, and carpane sustained platelet counts with no acute toxicity. That was in the Journal of Ethnopharmacology. Freeze-dried leaf juice was given to AG129 mice. This is a mosquito to vertebrate transmission model. Now, this showed no effect on NS1 and dengue viral mRNA, but it did show decreased levels of morbidity in the infected animals compared to control. And then freeze-dried juice again tested against AG129 mice. In this case, the researchers found increased plasma CCL2 and NCP1 levels. These are monocytes at the peak of viremia, so at the peak of viral infection. And this showed also a downregulation of inflammatory cytokine genes. So this showed a possible immunomodulating role. And that was published in the BMC Complementary Medicine and Therapies. Another leaf aqueous extract, this time in thrombocytopenia Sprague-Dawley rats. And they were able to demonstrate antiviral properties with the decrease of intracellular dengue viral load and preventing thrombocytopenia. And that was published in the Archives of Virology. So moving on now to some of the clinical studies. So these are studies in human beings. So the first one was using 30 mils of a fresh leaf juice extract. So literally taking the papaya leaf, juicing it and giving 30 mils once a day for 3 days. And this was an open label randomized controlled trial with 228 patients with both dengue fever and dengue hemorrhagic fever. The findings were a significant increase in mean platelet count in the intervention group compared to control. And that was in evidence-based complementary and alternative medicine. A standardized extract, so this is a nutraceutical developed with 1,100 milligrams of standardized extract. Initial pilot multi-center open labeled randomized comparative study. So a pilot study with 30 patients with both dengue and thrombocytopenia. So these showed significant increase in platelet count. And this was published in the Indian Medical Gazette in 2014. Then a 500 milligram nutraceutical tablet called Carapil, again, a leaf extract. This was a study in 400 patients with dengue fever and thrombocytopenia. They were given the 500 milligrams once daily, in addition to routine treatment as well. And they found a statistically significant increase in platelet count. So remember, when the platelet count is maintained and normalized, you do not see then people becoming hemorrhagic. So this was another one with the 1,100 milligram nutraceutical Carapil. This was a multicentric, double-blind, placebo-controlled trial. 300 patients tested across 5 centers. And this showed a platelet count statistically significant increase in the intervention group. And that was published in the Journal of the Association of Physicians of India in 2016. The same Carapil standardized extract given to 500 patients with dengue fever, again, statistically significant increased platelet count and a reduction in the average duration of hospital stay. And this one, the final one I'm going to present, is the standardized leaf extract in a syrup. And this is slightly different. This was actually a clinical trial, a prospective open label randomized controlled study in children. So this is a pediatric study, children aged 1 to 12. So the oral dose given was for children aged 1 to 5. They were given a dose of 275 milligrams 3 times daily, and the children aged 5 to 12 were given 550 milligrams 3 times a day. And again, statistically significant increase in mean platelet count in thrombocytopenia associated with dengue patients in pediatric patients. And that was published in the Pediatric the Journal of Pediatric Health and Medicine and Therapeutics in 2019. So we've seen different types of evidence all the way up to clinical studies. Now, what is underlying? What is it that causes Carica papaya to be able to work in this way? So the proposed mechanisms of actions include it causing membrane stabilization against dengue virus-induced peripheral destruction of platelets. It inhibits the dengue virus assembly. It prevents hemolysis and bleeding, and increases, very importantly, it increases the expression of the ALOX12 gene, which in turn elevates production of platelets. So here you see a diagram of Carica papaya leaf acting on the ALOX12 gene, increasing production of megakaryocytes that are precursors to proplatelets that then produce platelets. So it promotes the formation of megakaryocytes, inducing formation of platelets, and inhibits the lysis and destruction of proplatelets by the dengue virus itself. So in the summary for the proposed mechanisms of action are threefold. It's reported to act as an anti-inflammatory agent, to augment megakaryocyte development via the ALOX12 gene activity enhancement and subsequently platelet formation, and to exhibit direct antiviral properties. In terms of the safety, the biological safety reviewed from the clinical studies, it's been stated that the short-term consumption in adults is generally considered to be safe. Caution against use in pregnancy in people with liver impairment. The reported side effects from the clinical studies include mild GI disturbance and rash. And the potential herb-drug interactions, though not proven, would be with oral hypoglycemic agents, P-glycoprotein substance, and antibiotics. And therefore, those would need to be closely monitored. In terms of the dosage recommendation from the clinical studies, so these would be in standardized leaf extracts, the dose of 2.5 mil in children to 150 mil in adults, and a standardized tablet, standardized to 40% glycosides, 1100 milligrams 3 times a day. And this is the example in India of the nutraceutical that they have developed and which has been approved by the Indian authorities for the following indications: as an adjuvant for increasing platelet associated with dengue fever, and in thrombocytopenia for treatment and or prevention of bleeding. So the nutraceutical formulation is in both tablet form and syrup. The syrup is mainly aimed at treating children. Each of the tablets contains 1100 milligrams of a standardized leaf extract, and the syrup 275 milligrams per 5 mil, again of a standardized extract. So just to round off, I have focused very much on one plant because there is a very significant amount of research internationally to support its use. But there are other plants for which there is a growing body of research. 3 such plants include guava leaf, which is shown to improve platelet count, is antiviral, inhibits the formation of the dengue viral messenger RNA. Another plant known locally as rice bitters, Andrographis paniculata, has been shown to inhibit dengue virus. And neem leaf, also shown to have anti-dengue virus replication effects. So thank you. That brings to an end my deck. Thank you so much.
Thank you so much, Dr. Pickin. I'm so grateful for your presentation. Also happy that you were able to show that there were other plants, other medicinal plants that may have similar effectiveness in treating dengue fever and thrombocytopenia as well, associated with dengue fever. So thank you so very much for your presentation.
Thank you.
You're welcome. So we're going to move speedily along, and our next presenter is Dr. Sheena Francis. And Dr. Sheena Francis is going to talk to us about novel applications to override survival mechanism in mosquitoes. But before she does this, I'll tell you a little bit about Dr. Francis. So Sheena Francis is a senior lecturer and research fellow at the Natural Products Institute of the University of the West Indies, Mona campus. And her research, she investigates insecticidal resistance in mosquitoes that carry diseases that are of interest to humans. Her particular interest is in the identification of molecular markers associated with insecticide resistance in mosquitoes. She does this in order to elucidate the underlying bases of resistance in these insects. Her research studies have shed light on increased tolerance to insecticides by the Aedes aegypti mosquito in Jamaica. And she believes that understanding this more may lead to the development of compounds that can be used as insecticides, effective insecticides, and decrease the mosquito population. Of course, a reduction in the mosquito population would redound to a reduction in the diseases transmitted by mosquitoes. On the personal side, Dr. Francis is involved in multiple outreach programs aimed at fostering the creative minds of young adults and stimulating their passion in science and technology. Please join me in welcoming Dr. Sheena Francis to do her presentation. Sheena, over to you.
Good afternoon. Thanks so much for having me. So I'll be talking about novel applications to override survival mechanisms in mosquitoes. And here I'll be presenting some preliminary data on my studies. Just showing you a little bit about the work that I'm doing currently in the lab. All right, so as my talk suggests, I'm looking at mechanisms to override the survival mechanisms of mosquitoes. The mosquitoes the Aedes aegypti in particular, have become highly adapted to our environment. And you previously heard Dr. Webster talk about that. So they become efficient vectors. Aedes aegypti are the dominant vectors of dengue, and they only need roughly a cupful of water, as you heard our moderator say, to lay their eggs and start a new population in. So we live in homes and we have containers around us. These mosquitoes have adapted to breeding around our homes in small amount of water in containers, small containers that we might have lying down. They have adapted to the wet and dry seasons in that, as you heard Dr. Webster say, in the rainy months, any pooling or puddling of water, the mosquito can lay her eggs in and start a new population. In the drier months, we actually store water in containers. So again, we are providing a ground for our habitat for mosquitoes to lay their eggs in. You also heard Kerr spoke about the increase in temperature. The rate of development of mosquito has seemed to decrease with increase in temperature, in that a developmental stage from, say, egg all the way up to the adult stage of the mosquito usually takes 14 days under normal circumstances. However, in hotter seasons, that 14 is decreased to 9 days. The biting time has been varied in some countries based on our activities. Countries that tend to use insecticides in the day are observing mosquito and vice versa. Spring, they find that mosquitoes are biting outside of the house. And of course, we're in other countries where in the spring outside, mosquitoes are biting on the inside. Now, even the insecticides that we use, the mosquitoes have become resistant to that. So the insecticides that we're using to control the population of mosquitoes is decreasing in its effectiveness, because the mosquitoes have found a genetic modification to evade the insecticide, metabolizing it at a higher rate and driving the insecticides out of their body. So this issue is being seen globally. So this study was conducted between 2005 to 2016. So it's a little bit outdated, but notice on the map, everywhere that you see in blue are areas that reports Aedes species. And all those red and orange dots are areas where they have observed resistance to insecticides in the Aedes mosquitoes. I myself conducted some studies between 2017 and 2020, and I found that we do have mosquitoes in Jamaica that have become tolerant to insecticides, showing varying range of tolerance depending on the insecticide. So for pyrethroids, mosquitoes are very tolerant to those. For organophosphates, there's varying tolerance to those insecticides. So basically, what can we do based on to decrease mosquito population, based on the fact that these beautiful mosquitoes have adapted to our environment to live comfortably with us? So in Jamaica, you know, it's a tropical country. We're surrounded by loads of plants. You just heard Dr. Pickin talk about a wide variety of plants that can be found here in Jamaica that have effects against the disease itself, dengue. So can we use plants as insecticides? The answer for that is yes. A study conducted by Cantrell, a review paper presented by Cantrell in 2012 showed that over 35.7% of the pesticides, broad spectrum pesticides to treat all insects, both agriculture and disease, insects of human concern, are derived from natural products. So with this, in a lab currently, I am looking at plants, Jamaican plants, that have effect against the mosquitoes, that have a toxic effect against the mosquitoes. So I've coded the plants because it's ongoing studies, please forgive me, but I have 3 different treatments that these plants are in. So there's treatment 1 in blue, treatment 2 in red, and treatment 3 in gray. And depending on the plant and depending on the treatment, some plants, only one particular treatment actually works. Other plant, none of the treatment works at killing the mosquito. This shows percentage kill. Other plants, all 3 treatments is killing the mosquito. So what I'm doing now in the lab is finding out what component of the plant is responsible for the toxic effect and how is it killing the mosquito. So I'm trying to find out what is exactly and how it is doing it. So hopefully if you continue to follow my research, you in the near future, I hope to present some of the findings of these studies. The other study that I am recently involved in, and Dr. Antoine will be telling you more about that, is looking at sterile mosquitoes. Now, the female mosquito, the female Aedes mosquito, is the main mosquito that is responsible for transmitting the dengue virus. And that particular sex is responsible because the female feeds on blood. She's the one that bites us. She's the one that imbibes our blood. And she needs the blood to metabolize and produce her eggs to develop a new population. So it's the female mosquito that we're really interested in when we're saying decreased population of dengue transmitting mosquito, the female Aedes aegypti. So my study looks at competition between the mosquito population. Now, sterile insect technique has been used on many other insect populations other than mosquitoes. However, it's been shown to be quite effective at decreasing other insect population. However, its application in mosquito is relatively-ish new. And there are some challenges. So researchers like myself are working on combating some of these challenges, dealing with them and seeing how best to enhance the technology or the technique. So I am particularly looking at survival of the sexiest mosquito. So as I said, it's the female mosquito that transmits the disease. So if we sterilize the male, we want to see how the male, the sterile male competes with the wild male in the environment. So for this experiment, I have, it is in its very, very initial stage. So I'll be looking at the rearing of the mosquitoes, the morphology and survival of these mosquitoes, their pre-copulatory behavior, and their mating success. And I'll be varying the treatments throughout each to explore how successful these male mosquitoes are at competing with the wild population for a wild female population, for a wild female mating. So here ends my talk. And if you have any questions for me, let me hear after the talk. And also you can email me and follow us on our website, www.mono.ue.edu/NPI, to find out our progresses. Thank you very much.
Thank you so very much, Dr. Francis. That was a very good presentation and quite succinct. I'm sure there's a lot more that you could have said, but you managed to keep it together in time. So thank you for that. And we're looking forward to the questions that will come to you at the end of this session. So I'm going to move on to our next speaker. And if you were not here earlier, you may not have heard that Professor Charles Grant had a personal emergency and had to leave. And so in very quick order, Mr. Johan Antoine agreed to fill in for Professor Charles. And as I said earlier, I think Mr. Antoine is an excellent replacement and alternative. And he's quite aware of what Charles was going to present. So we're looking forward to the presentation as well. So I'm going to introduce Mr. Johan Antoine very quickly. Mr. Johan Antoine is a research scientist at the International Center for Environmental and Nuclear Sciences, ICENS, where he has been for over 19 years. He is a section head of the Nuclear Analytical Laboratory at ICENS and an operator of the Slowpoke 2 nuclear reactor. He's an author of several peer-reviewed journal articles, conference publications, technical reports, and 2 book chapters, as well as a frequent reviewer for several high-impact international journals. Mr. Antoine's expertise is in neutron activation analysis, and he has acted as a consultant and expert for the International Atomic Energy Agency in this capacity. He has also had roles as chief scientific investigator and project counterpart for several IAEA projects. His current research interest is in the use of nuclear analytical techniques in the field of forensic science, but he previously used neutron activation analysis for food research, health risk, and toxicology assessments, and also for environmental studies. Mr. Antoine, thank you again for jumping in at the last minute, for being a great team player, and we look forward to your presentation.
All right. Thank you very much, Dr. Gordon-Strachan. What I'm going to talk about in lieu of Prof, who unfortunately had to be away for an personal emergency, is actually the sterile insect technique. So we're going to talk a bit about how we're using radiation and a specific type of radiation to be a form of mosquito control, pest control in this case, and the part that iSense intends to play in this larger process of mosquito population control. All right, so I'm going to go through this very quickly. Brief introduction to the type of radiation we want to talk about, the effects of that type of irradiation, the uses of ionizing radiation, which I won't spend too much time on, the sterile insect technique, which I will spend a little time on, the advantages of this technique, the iSense facility that we're going to commission, considerations for the successful implementation of any SIT program, the conditional approach that is necessary for that success, and a little bit on the conclusion. All right, so we can break up radiation and the type of radioactivity we're going to talk about into ionizing and non-ionizing radiation. In the graphic that you can see here, you're looking mostly at non-ionizing radiation, so all the way up to ultraviolet. Ultraviolet is non-ionizing radiation. And so that's not the radioactivity that we're looking at for the purposes of the sterile insect technique. So X-rays, gamma rays, alpha particles, beta rays, those are ionizing radioactivity. Those have the energy and are energetic enough to ionize molecules. It is X-rays and gamma rays specifically that we're looking at when we're considering the sterile insect technique. So gamma radiation in this case, what we want to bring is gamma radiation, that form of electromagnetic energy that will create the ionization that we're interested in at this point. All right, so in the case of chemistry, physics, where we can convert electrically, we can convert neutral atoms or molecules into electrically charged atoms. We need a certain energy. In the case of X-rays and gamma rays, for our purposes, they're exactly the same. What we are considering in this case is where they originate from. But for our purposes, the gamma rays originate from the atomic nuclei, and the X-rays originate from the shell or orbital. We tend at iSense to favor gamma rays, and I'll explain that in a bit. But the situation here is that what we are looking for in this effect, and I spend a lot of time talking about how we can respect radioactivity and ionizing radioactivity without it creating any harm to the people who utilize it. In this case, what we really are looking for now is to actually create a certain type of damage. We're trying in this case to make it for our beautiful mosquitoes, as Dr. Francis mentioned. We're trying to create a specific type of damage, and I use the term specific not loosely. It has to be entirely specific to achieve the goals that we want to achieve. All right, so use ionizing radioactivity for many things, including sterilization. We use it for food irradiation. We use it in medicine. We use it in industry. We use it, and I want to use it when we do actually commission this facility for other aspects other than the sterile insect technique. I want to look at phytosanitation. I want to look at food irradiation. I want to look at sterilization of skin grafts for burn victims, medical equipment. But for now, we're going to stick to the matter at hand. All right. So, what is the sterile insect technique? The sterile insect technique is a form of birth control for mosquitoes. We are actually, as Dr. Francis mentioned before, introducing sterile male mosquitoes to our population, and there's certain steps in that process that we have to take before that is introduced to eventually reduce the population, either for maintenance or eradication. In this case, the policy at this point is eradication, because we're looking at all the diseases that are carried by the Aedes aegypti and potentially the Aedes albopictus mosquitoes, and we're looking to eradicate that circumstance. So what I want to show with this graphic is really important. The sterile insect technique is still only a portion. If you're looking towards eradication and you look at that graph, you'll see the green part of that graph. That's the sterile insect technique. You still have to have it as a part, maybe the knockout punch, but the last part of an area-wide integrated pest management program. So if all the other things that we have spoken about, including unfortunately what Dr. Francis mentioned about the about mosquitoes being tolerant and resistant to some of these insecticides, those aspects have to work before the SIT can not only be introduced, but can be expected to be successful. All right, so a little bit about the SIT. The sterile insect technique is pretty old, right? The concept was developed in the 30s, and there were several researchers who were working on it at the same time and came up with the same aspect that, hey, we can induce sterility, and specifically those dominant lethal mutations that can result in the sterility of insects. And it was first tried on the New World screwworm fly, and it had great success. Several researchers could then show that this had an economic basis, it was an economic success, and it didn't take long before people could see its applications throughout the world and throughout various other agricultural aspects. And so, although it's not particularly new, it has not necessarily been exploited to the level that it could have been exploited to. And in certain cases, there have been, as Dr. Francis mentioned, some setbacks. But in the case of diphtheria, those are flies, you've had a wide range of whether there has been success or failure, as has been the case with Lepidoptera, those are butterflies and moths, my apology, that can actually have economic damage to various ventures. All right, so focus here. The trick here is that what I'm trying to do is to be very, very, very specific about the dose that I'm exposing my mosquitoes to. So the dose should be enough that when I have exposed that mosquito, that male mosquito, ideally, to a dose of gamma radiation, that in an attempt to repair the damage that has resulted from that exposure, the repair is going to be, let us call it, incomplete. It's going to be incorrect. And when that happens, it's going to lead to a dominant lethal mutation. That is particularly important, because what I need that to do is not to stop the process of fertilization and not to stop the process of development up to a certain point. What needs to happen is that it needs to be competitive. So it cannot be absolutely dead. The sperm from the male has to be competitive enough to reach the development of a zygote, but not so for an embryo. So the competitiveness of the sperm is absolutely important in this, and that's a theme that we're going to come back to over and over again: the competition. All right, here's a nice graphic on the effects of radiation, an increasing dose on the ovary length of Aedes albopictus, and you can already see that. And this is not quite for the purpose of SIT, It's more to show the effects, the biological effects, and the damage that can occur from exposure to increasing doses of radioactivity. Because at the end of the day, what I'm looking at, if I were to show you 0 and to show you 50, if what I was doing for the female albopictus in this case was to ensure only a certain level of fertility, to prove my point here, I would have to be looking not at the highest dose, but something more in the middle, something just enough to make her competitive, just enough for the process to continue, but not enough for it to reach a successful mating. All right, so here's my job. My job, if we look at that graph, so what you're seeing is a sigmoid curve. And the dotted line is competitiveness, right? The unbroken line is fertility. And what I'm trying to do, ideally, and what our research will primarily focus on, is making those lines, those sigmoid curves, as close together as possible. Because what I want is maximum competitiveness with the lowest amount of fertility. That's what dose optimization, which is what iSense is going to be primarily involved in, is going to be researching and achieving. That point where we can be sure that that male mosquito, having been irradiated, having been made fertile, is still competitive enough so that when we release it, it can compete with wild male mosquitoes. If I release underdosed males, then they're fertile. And all I've done is release 500,000 to 1 million individuals to increase the population. And that's a disaster. On the other hand, if I overdose, then what my males may look like is, well, you see me? I'm in a suit. Maybe I look okay. Maybe an irradiated male, an over-irradiated male might look like his suit is falling off, it's torn, it's disheveled, and will not likely be able to compete properly with the wild type. The female will look at that mosquito and say, something is wrong, and be not interested. The likelihood of the female selecting that male is going to be less. I need my mosquito to look like nothing has happened to him. And so he has an equal chance of being selected by the female. So dose optimization is very, very important. Make that male fertile, but make him still look like there's nothing wrong with him. He's as attractive as the male that has been unirradiated. All right, so a graphic that simply shows where we fall in this. The mass rearing is not at iSense. The release is not by iSense. What iSense is going to be doing is exposing those mosquitoes to the dose, ideally, that will make them competitively sterile, or vice versa. What we want is sterile males so that the MOHW can then release those males and compete with wild males. All right, so what are the advantages of SIT? So, sterile insects are not self-replicating. They cannot become established in the environment because what we're doing is reducing the population. It is autocidal control because it is species specific. I'm only irradiating a specific species. It's not affecting any other. It's not affecting, in other words, if I irradiate Egypti, I'm not affecting albopictus. It is species specific. It does not introduce, and our moderator mentioned that before this began, it doesn't introduce non-native species. It is using the very species that we want to reduce the population. There's no use of chemicals, there's no residue to affect the environment, and there's inverse density dependence when we are using the SIT. So it works best as the population goes down. So as we reduce the wild population and add more irradiated males, it further reduces the population, and we add more irradiated males until it brings it down to, as I said, maintenance, or in the case of what we really want, eradication. All right, so it's not quite the ISENS facility, but what we hope it will look like to a certain extent. This is the exact same irradiator. So it's going to use a cobalt source, and we are going to actually by just to give a little bit of information about it, by its footprint, it's really very small. It's about 3 feet across, it's 4 feet up, not too much more than that, about 4 feet high. But it weighs about 17,000 pounds, mainly because of the shielding, because it is totally self-shielded. And of course, it has a lot of heavy metals in it in the case of cobalt 60. So between the shielding and the source itself, it is very, very heavy. All right, now this is a little bit more of what ISENS does, and I'm just going to tell you about how it comes about. What ISENS does is a lot of its neutron activation analysis. You have a neutron, it's known as a thermal neutron, so it moves slowly. And I use that to hit a uranium-235 atom, which is our fuel, the fuel that we have in the reactor. And it usually means that this is going to release, on average, 2.3 neutrons. That uranium atom is going to split up into any number of things. We like to show in this graph that it might be barium and krypton. It can be any number of elements. But what it's going to release, which is more important, is our neutron. And when we bombard either cobalt 59 or nickel 60, we are going to end up with cobalt 60. And cobalt 60 has very, very energetic gammas at energies of 1.17 or 1.3 mega electron volts. Very, very powerful to do exactly what we want to do in this case. So let's consider a successful implementation of any SIT program. 1, the species must produce exclusively sexually. Obviously, if it's asexual production, if there's parthenogenesis, then having a sterile male, even a fertile male, doesn't matter. It's going to defeat the entire purpose. There must be a mass rearing facility. Procedures must be available. Those are already the case. We already have the MOHW up to between 500,000 and 1 million mosquitoes a week. So we already have that in place and locally in place, which is also very important. The released insects must be able to disperse among a certain amount. You don't want it entirely dispersible so that you put it at one location and it's gone all over the place, or else the effect is dilute. But at the same point in time, if they're extremely localized, you defeat the purpose as well. As I mentioned, remember that sigmoid curve. Sterilization is of no use if the mosquito, the male mosquito, cannot or will not be selected. It cannot compete. It doesn't mate. Then it doesn't matter, because the female will always choose a fertile male. The females must only mate once. If they can mate again, then there's also a likelihood that she can mate with a fertile male. And then again, the purpose is defeated. And remember what I mentioned about that irradiated sperm. It must be competitive enough to reach the point of a zygote. And again, the population density must be low. So remember that graph that I showed before with the blue and the green, where the SIT represent the green aspect of an area-wide integrated pest management program. If it doesn't have a before that reduces the population, SIT will not work. All right, so phase conditional approach. And this is another aspect of the success of the SIT. If there is not a lot of research, a lot of preliminary data collection, especially about population dynamics, including the size, then again, introducing SIT too early or without this critical data is going to likely result in a failure of the process. And this kind of expands what I've shown you, where there is continual trial, continual processing of data, and continual analysis of the results before you can even reach the point of releasing these sterile insects into let us even say a wild population outside of any laboratory considerations. All right. So it is not a silver bullet. It is not the thing that is going to eradicate any mosquito, or for that matter, any insect of its target without another aspect of or pre-aspect of a program, especially in the case of mosquitoes. I'm not going to be producing radioactive males. I am not going to be making a mosquito that flies around. It's not going to be radioactive. There is no introduction of transgenic animals. There's no introduction of any cross-species aspect. Everything remains within species. That graph that I showed you previously has to be followed for the process to be complete and successful. And I like to say maybe it involves a combination of sterilization techniques, for instance, insect incompatibility techniques. I don't want to get too much into that because I believe that this could be successful just as the radiation-based sterile insect technique, to be honest with you. All right, thank you for that.
And thank you. And thank you so very much, Johan. We would not have known that you did not prepare and present this yourself. You did an excellent job. So thank you so very, very much. I'd like to take this opportunity to thank all our speakers. They were brilliant, excellent, and I learned so much during this session and during these presentations. So we have a few questions. And I'll start off with a question that came from the audience. And this question is directed to Dr. Webster-Carr. So the question is the country at the point where we can predict outbreaks since we know the causative factors? And if yes, did we anticipate this outbreak?
We did, in a sense, anticipate the outbreak. It's just really, I don't even want to say the word predict, because some like we do fancier things that we do. But based on the periodicity that we have been observing, we would expect that to happen. And given what we had earlier this year with the increased temperature, I can't remember days you said the highest temperature ever or whatever. And we had a number of days that were above average. So with that, we said, it's possible. In addition, when we got our first set of dengue confirmed cases back in July, and it was type 2, all were type 2. We said we haven't really seen type 2 predominating since 2010. So that was an increased risk. And this is why we had put the country on alert from when we got back those results. So, yeah.
So just to follow on that question, I know that we've been talking about setting up dengue fever early warning and response system that would use statistical modeling to project, maybe not predict, but project the likelihood of outbreaks. I just wondered if you would want to comment on that.
Yeah, so the good thing is that we now have After completion of this year, we'll have 10 years of data looking at on a weekly basis. So right now, we're really delayed in our identification of alert and outbreak because we have monthly
The data.
yeah, but now at the close of this year, we'll have 10 good years of weekly data and daily if we look at it. So we can do, we can do a lot of things with that. So hopefully we'll get to that.
Okay, so we have a few students who are looking in that area, so maybe we will have a chat afterwards. So thanks, Dr. Webster. Thank you for your response. Another question came from the chat, from the question and answer portal. And this question is to your hand, Mr. Antoine. So the question is, how many mosquitoes would have to be irradiated to really make a difference? And there are some follow-on questions. I'll ask those at the same time. Where will we get them from? And can anything else go wrong with the mosquitoes when we expose them to radiation? Could we be creating a bigger problem? So if you want, I can break it down and then you can answer section by section. So the first section was, how many mosquitoes would have to be irradiated to really make a difference?
Okay, good question. That depends on the population dynamics. However, based on preliminary estimates, I mentioned that during my presentation, you would need about about 1 million a week to release about 1 million a week. So 1 million irradiated mosquitoes a week to start making a difference. And again, recall that we also need to see a certain level of reduction in that population before even that million would matter. You're looking at 1 million after considering that you may have 10 million mosquitoes in a given area, in the type of area where that million would actually make some kind of difference. You'd have to bring that down significantly before that million would even matter. So that's the first aspect. You can remind me of the second question, Dr. Gordon-Strong?
Sure. Okay, so the next question is, where would we get that million from?
Okay, so you literally just breed. You just breed those mosquitoes. You can capture and then start rearing from your wild population. So it's not that we're just grabbing something off nets or something like that, but in a way, yes, we are. We're just collecting, and then you start your breeding process. So that is why, as I mentioned, you have mass rearing facilities that do exactly that. Those facilities then come to a place like iSense for the irradiation aspect, and they're returned to the facility, who then will coordinate the release activities.
And this is done under very controlled.
Absolutely,
okay.
Absolutely. And what can go wrong? Well, remember that phased conditional approach. The truth is, what is most likely to go wrong is that you don't have the initial, what we call conventional reduction of the population. So everything outside of SIT. And so you release perfectly fertile competitive mosquitoes and nothing happens. It becomes a nuisance that you see because, as Dr. Francis mentioned, it's not the male mosquitoes that are biting, but you're going to still see an inundation of mosquitoes. What can be worse than that is if our dose optimization is not up to par and the conditional approach does not spot that we have a certain level of underdosed mosquitoes. And that means that we are releasing fertile mosquitoes. So instead of releasing mosquitoes that can help reduce the population, you're actually increasing the population. What is more likely is that we overdose in an attempt to ensure that those mosquitoes are fertile. And instead, are in fact are sterile, sorry. And instead we get no competitiveness. That's likely. That is a likely error. And therefore it doesn't work.
Thank you. Thank you, Mr. Antoine, and thank you, Dr. Webster-Carr. We have a few other questions. I want to pick up with Dr. Pekin, and I want to thank you for your presentation. But I wanted to just ask you a little bit about some of the barriers to developing and commercializing nutraceuticals in Jamaica, because it seems to me that you have an extract that has shown its effectiveness all over the world. And we do have Papaw Grows Wild here. So why is it that we're not able to move from extract, or what's holding us back from moving from the extract to the commercialization of this as a nutraceutical?
I think the main issue probably affecting us at the moment is that we don't yet have in place a regulatory framework for nutraceuticals. So in Jamaica at the moment, we have the Food and Drug Act. So something's either classified as a food or it's classified as a drug. There's no in between. And so we had a committee that actually ran for 3 years that was set up. It was run under the National Commission for Science and Technology, led by the Ministry of Health. And that actually came up with an amendment to the existing Food and Drug Act that would recognize natural health products. So that was submitted. So we're waiting basically for that legislation to go through. That sits with the minister. And that was sent to the Attorney General's chambers for review back in July 2021. And that would give us something similar to the Canadian system, which recognizes natural health products and puts in place a regulatory framework that would allow those products to be quality assured and managed through to utilization within the health system. So doctors would be able to prescribe, et cetera, et cetera.
And just picking up on that, the question of dosage, sometimes it's said that with nutraceuticals, the dosage is hard to determine. I remember that your presentation spoke about dosage. And is that standardized, and would we be able to utilize that dosage, or would we need to do additional research on our own papaya leaves to come up with a dosage regimen?
Yeah, I mean, the challenge with natural health products is where it's grown, the growing conditions, you can get significant variations in the levels. I mean, I think my first point would be to say, with the model of Tramil, we've got a dengue outbreak right now. I think traditionally people have used medicinal plants from their yards. So I would say, based on the research, people can draw from that research and actually use what they have around them in order to stay well and prevent themselves getting into hospital situations. But moving on from that, yes, we can develop a standardized extract from the plants grown here in Jamaica. And, with the right funding and with some nice funded projects, we could get those products onto the market quite quickly, I believe. And because the clinical studies, and some people might criticize the quality of the clinical studies, but the data's there. There's a significant amount of research that identifies what is efficacious and what's safe. And therefore, moving to a standardized product should be pretty straightforward.
All right, thank you. Thank you so much, Dr. Pekin. I'm going to double back to Dr. Webster-Carr, and just wanted to ask a quick question about dengue outbreak serotype 2. In your presentation, you said that mostly children are affected, children 5 to 14, 1 to 4, and under 1. And you alluded to the suspicion, or maybe the fact, that the last time we had a circulation of serotype 2 dengue, it was in 2010. So children now would not have been exposed to that virus. I wondered if you could tell us a little bit about whether or not type 2 has a different kind of presentation, because it seems to be associated with more hospitalizations. And I wondered if there is something special about type 2 that's causing the level of hospitalizations and so on that we're seeing here in Jamaica at this time.
So when you look at this, some studies say yes, type 2 has more severe disease than others, but others say differently. What is more indicative or can predict greater level of severity is the history or having had another dengue serotype that increases your risk of severe disease. So since 2010, when type 2 last predominated, we have had predomination of all the other types. And so are some of these secondary infections, and hence why we're seeing more severe disease. So if I had to come down on one, I'm thinking it is more, probably a more secondary infection than per se type 2 being more severe.
All right, so just to add on to that, since then we've had chikungunya and Zika, and I'm wondering if the exposure to those viruses and diseases as well could have made just like you're saying, made this outbreak a bit worse, because it would be kind of like a knock-on effect from having a previous exposure to vector-borne arbovirus.
So I would, chikungunya is probably out. Anything is possible because Zika is in the same family as the flavivirus, as as dengue, and they do cross react. So do they give some protection or do they cause more severe? I don't know. But
okay, all right. So that is some research to be determined. Okay, I want to just cross over now to Dr. Sheena Francis. And Sheena, I have one question for you, and that question is looking at insecticides. Now, some years ago, I was in a meeting with you with the USAID, and we were looking at how effective insecticides were, and especially insecticides that were developed from natural products. I know that Brazil, for example, has been working a lot on this, and on the global front, work has been done on this as well. So I wondered if you could tell us a little bit about what's happening in Jamaica and how that experience, maybe the Brazilian experience, might be beneficial to what we're trying to do here in Jamaica at the moment.
Okay, thanks for that question. I appreciate it. So the use of plants as insecticides is by no means novel. A very predominant class of insecticides, the pyrethroids, was actually developed initially from a plant, the chrysanthemum, right? So the use of plants is not novel, and the plants application as insecticides are increasing. Broad spectrum, not just treating for mosquito, but even for agricultural pests. And in Jamaica and the wider Caribbean, we're looking at, our researchers like myself are looking at the plants and trying to scope out plants and the particular, looking at their toxicity and looking at the specific chemicals within the plant that actually make them toxic to the mosquitoes. And we're looking at how it's working. So the thing is that when we're presenting the data, a lot of, if you comb the journals, there are a lot of information about plants having insecticidal activity. The thing is that we haven't as yet found the plant that supersedes insecticides that are on the market. So we have to try and find a plant with this toxic compound that is more toxic than the insecticides that are currently available. And the second thing, we have to be able to tell how this new compound is working. And then it has to go through several environmental tests after that before it can reach to market level. So we're in the stage, at least for my lab, I'm in the stage where I'm just looking at how the compounds are working, how they're interacting with the mosquitoes, and to say this is how it's killing the mosquitoes. So once I have that information out, then the next phase would be to go on to say, is it toxic to the environment? How toxic is it to non-target species like fish, crabs, frogs, that sort of thing?
Okay, because I was going to ask you, that was going to be my next question, which is, how do you know how toxic it will be to the environment within which it's used, male mosquitoes, and we are able to have the population under control to the extent where it may become extinct? What might be some of the natural or ecological challenges that we may face, and how would this affect our natural environment? How might this affect us?
All right. Thank you for that question, because it is something that has come up when I've delivered talks such as these before, where people are concerned that we're removing an integral part of the environment. And the first thing I remember that an entomologist who was delivering a lecture with me mentioned is that there are dozens of other mosquito species. The removal of that species, in the case of Aedes aegypti, is really not going to have the type of effect that we believe it will, simply because we're not removing 1 out of 3 or 4 mosquitoes. We tend only to think of mosquitoes as, for instance, the Aedes species, Culex species, Anopheles species, and so on. What we end up with is thinking that there are only 3 or 4 species of mosquitoes that are in our environment, when they're more like 75, I think, if I recall my biology. Don't hold me to that. But there are many, many more species that are actually far more ecologically active than Aedes aegypti, or in the case of the tiger mosquito, Aedes albopictus. So that aspect, removing that species, is not going to have the type of effect that we think it will in terms of ecologically speaking. What feeds on them will continue to feed on other mosquitoes because they're there in the environment, right?
I want you to remember that mosquitoes are also pollinators, and especially
precisely so.
Pollinators. And just like we're trying so hard to increase our bee population to ensure that we have pollinators, how do you know for sure? Or can you say?
Because what you're saying is that the niche that they have in that respect can be taken on by other mosquitoes, right? Okay. And actually other insects in certain cases, right? So the gain, the pro of removing them is far greater than the loss in this case.
Okay. All right. Thank you. Good response. Okay. All right. So I'm going to have to go back to Dr. Webster Carr, and I'll ask her to forgive me. I think my own work I've done some work in dengue fever, so her presentation was quite interesting to me. And I just have one question about, again, the children who are being infected and exposed. And I wondered if you could speak a little bit about whether or not the Ministry of Health has embarked on a school program to do fogging and to certainly reduce the mosquito population within the school environment. I wondered if you would make a comment on that. Dr. Webster, you might be muted. Dr. Webster, you're muted.
Sorry, I got distracted. I got a call. Sorry, could you repeat? I apologize.
Oh, that's all right. What I was asking was about so the children who are getting this illness, they're mostly children of school age. So I wanted you to comment on the government's attempt to reduce the mosquito population within the school environment. I'm sure this is a target area, so I just wanted you to make a comment.
We saw the age group. We met with the Ministry of Education, and we went and did the search and destroy activities, and like weekly fogging of the schools, and for children, once they have been identified, not to expose the others, or increase the risk of biting and transmission after a few days. But we're trying to, for this outbreak period, to fog like on a weekly basis as best as we can to interrupt the that cycle, especially in the schools. Over.
Thank you, Dr. Webster-Carr. I think I just need to check in for one second with Terry. I think we're almost at the end, and I'm just searching through to see if there are any other questions online. I'll just have a very quick look. And so far, I'm not seeing any. So I wanted to just maybe close off the session by personally thanking our presenters for excellent presentation, for an excellent afternoon, for really telling us a lot about some of the ways that science is working to try to reduce the mosquito population and to reduce the outbreak of dengue within our population. And so it is my pleasure on behalf of the Faculty of Science and Technology to thank you all for giving us of your time. You know, time is a non-renewable resource, so we treasure the fact that you chose to spend time with us. So for those presenters, for our presenters, thank you very, very much. And for those who joined online, we also thank you. I think you will agree with me that this was time well spent. So it's over and out, and have a great rest of the day, evening, night, wherever you are in the world. Thank you.